So we've been talking about general types of reproduction, starting off with asexual reproduction, and more recently talking about sexual reproduction. And so I want to continue talking about sexual reproduction. As a quick reminder, in the previous slides, we discussed that sexual reproduction requires meiosis and fertilization. And importantly, this process of fertilization doubles ploidy. So you go from two haploid gametes joining together to make a diploid zygote. And this process of meiosis, importantly, it halves ploidy. So it cuts the ploidy in half, where you go from a diploid zygote to haploid gametes. And also, if you're dealing with a multicellular organism, this diploid zygote these cells will create copies of themselves to go from unicellular to multicellular organism. And it will use the process of mitosis, which maintains the same ploidy. Okay, so all this stuff should be a bit of a review for you. If you've not watched the previous slides, please check those out in the previous lecture. And if all this stuff has been a while since you've looked at meiosis and mitosis and things like that, I highly recommend you review those processes in your textbook. Okay, so I want to remind you of the relationship between ploidy and variation in traits. And please remember that we care about variation in traits because variation in traits is an important requirement for natural selection. You've got to have variation in traits in a population for natural selection to choose from, okay? So let's pretend we're looking at some imaginary chromosome number 8. And so this is a diploid organism that has 2 copies of chromosome number 8 in its nucleus, okay? And somewhere on chromosome number 8, there is a gene for beak depth, okay? So how thick your beak is if you're a bird. And remember that because this organism is diploid, it can have different alleles or different versions of that same gene for beak depth on the different chromosomes. So it could have one allele for a deep beak and a different allele for a shallow beak. Okay, and so having multiple chromosomes in an individual cell allows for different combinations of these 2 alleles. And those different combinations of alleles, in turn, can result in different phenotypes, or what that genetic trait looks like. So for example, you could have an individual with 2 chromosomes that have little b and little b alleles, and that individual will have a short beak, or you could have an individual that has 2 big B alleles in its 2 chromosomes and has a very deep, thick beak, okay? And so having a diploid organism, having ploidy, allows organisms to have multiple alleles, and those different combinations of alleles that give rise to different traits or phenotypes in the population, and ultimately more variation in the population for natural selection to work on. So that's important. So as another reminder, remember that mitosis is the only process that's used for asexual reproduction in eukaryotic organisms. So anytime a eukaryotic organism reproduces asexually, it's always going to be through mitosis. And there is no chance for any new combinations of alleles in any mitotically produced cell or organism. Those daughter cells are always going to be genetically identical to the parent cells. By definition, mitosis produces genetically identical daughter cells that have the exact same combinations of chromosomes and the exact same combinations of alleles in every single daughter cell. And so as a result, asexual reproduction is going to reduce the overall genetic variation in the offspring because the offspring are going to be genetically identical to the parent, okay? So this is in contrast to sexual reproduction, okay? So just as a reminder, asexual reproduction, the offspring are genetically identical to the parents unless mutation occurs, and that's because mitosis is used. And so there's very little genetic variation in a population that's specifically or only reproducing asexually. And this is in contrast to a population that's reproducing sexually. So sexual reproduction can actually create a lot of genetic variation where offspring are genetically unique from the parents because meiosis and fertilization are involved, okay? And so fertilization, remember, is this process where 2 haploid gametes come together to produce a diploid zygote, okay? And so fertilization can actually make unique combinations of chromosomes and therefore unique combinations of alleles, and therefore these diploid cells that are produced can be genetically unique from their parents, okay? Now, in addition, this process of meiosis can also make unique combinations of chromosomes and therefore alleles. And that results in new combinations of chromosomes in these haploid cells here that can ultimately result in new combinations of alleles and differences between parent cells and the offspring cells that are produced. Okay, so let me go into this in a little bit more detail for how fertilization meiosis in sexual reproduction result in lots of genetic variation in the offspring. Okay, so let's first talk about meiosis. Okay, so meiosis involves a couple of important processes, specifically independent assortment of chromosomes during meiosis 1 and something called crossing over of chromosomal material during meiosis 1, okay? And these two processes are really, really important for creating genetic variation, as is this other process of mixing gametes between unrelated individuals during fertilization. So I'm going to break down what's going on in each of these individual processes to create genetic variation that's needed for natural selection to work and variation in the population. Okay, so let's first start with this idea of independent assortment. Okay, and basically the idea of independent assortment basically says that genetic makeup of gametes depends on how chromosomes aligned back in meiosis 1. So if we're looking at the entire process of meiosis from start to finish, where you're starting with a parent cell over here and the haploid gametes that are produced ultimately over here, there's a couple processes that happen in between, where essentially you're starting with copies of these individual chromosomes. So we can call this chromosome number 1 here and the two different versions of chromosome number 1, and then this is chromosome number 2 here and the two different versions of chromosome number 2. And so the copies, so chromosome number 1 and 2, can just by chance align this way in meiosis 1, and then they'll get split apart once, and then they'll split apart again to make this split apart here and here again to make these haploid gametes that have the full complement. So it has chromosome number 1 and chromosome number 2, chromosome number 1, chromosome number 2, chromosome 1, 2, chromosome 1 and 2. It has the full complement of chromosomes, but only one version of each of those. And just by chance, the haploid cells end up looking like this based on how things split and how things initially aligned right here. But imagine this alternative scenario where just by chance, instead of aligning with the reds on one side and the blues on one side, they happen to align like this, with the reds on opposite sides and the blues on opposite sides. So you still got the full complement of chromosomes where you've got chromosome 1 and 2 here, 1 and 2 here. It's just that now the different versions of those different chromosomes have aligned differently. And as a result, when this cell splits, you get cells that look like this in the next stage of meiosis. And then when these cells split, you'll get this combination of gametes. And so what you can see here is there's still the full combination of chromosomes in each of these gametes, where you've got chromosome 1 and 2, chromosome 1 and 2, chromosome 1 and 2, chromosome 1 and 2, but you've got different combinations of those chromosomes, so different alleles on these different chromosomes in each of the gametes. So here you got the blues all combined in the gametes, here you've got blues and reds combined in the gametes, okay? And so this is this idea of independent assortment, where here these chromosomes assort independently of one another. That can result in different alignments here and ultimately different combinations of chromosomes or different combinations of alleles in the gametes. Okay, so independent assortment creates unique combinations of chromosomes in the gametes while ensuring that one copy of each chromosome is always present. So you've always got the right number of chromosomes. You've always got chromosome 1 through whatever. But the versions of those chromosomes that you have are different in the gametes. Okay, so I know this can be very confusing for folks, so spend some time kind of thinking this through and really wrapping your mind about what this means. But ultimately, the important thing here is that independent assortment can create unique combinations of chromosomes in the gametes. So you can potentially get this combination, or you can get this combination here, depending on how these chromosomes independently assort at this step in meiosis. Okay, so how much variability can this independent assortment of those homologous chromosomes, of those chromosomes at that step in meiosis, produce? So as it turns out, a parent cell can produce 2 to the n possible gametes when n equals the haploid number, okay? So independent assortment can produce 2 to the n possible different combinations of alleles in different gametes, where n equals the haploid number. So for example, in humans, n is the number of chromosomes, so n is 23. So that means there are 2 to the 23rd or 8.4 million different genetically unique types of gametes that can potentially be produced by one human just by this independent assortment of the alleles, or I'm sorry, independent assortment of the chromosomes during meiosis. So if this is the parent cell, meiosis can produce 8.4 million different combinations of these 23 chromosomes, depending on whether you get this version of chromosome 1 or that chromosome version of chromosome 1, or this one versus that one of 2, so on and so forth. You can get 8.4 million different combinations of these 23 different chromosomes because of independent assortment, okay? And if you add in mixing of gametes from other individuals, so in other words, a sperm or a father's sperm can have 8.4 million different combinations, and the mother's egg can also have 8.4 million different combinations of her 23 different chromosomes. If you multiply this times this, so you get 8.4 million different potential combinations here, 8.4 million different combinations here, multiply that times that, you can get 7 times 10 to the 12th different possible combinations from just one mating, because there's 8.4 million from different combinations from the mom and 8.4 different million combinations potentially from the dad. So this is a huge amount of variation, and this is a big part of the reason why siblings look different, because you can have so many different combinations from this independent assortment that the combination you might get for your sister or brother is going to be different than the combination that you get for you and your next brother or sister, okay? So who cares about genetic variation? Why do we care about this? Remember that genetic variation is the raw material for natural selection, all right? And so you need to have this variation in the population. And if this wasn't enough opportunity for genetic variation during sexual reproduction, there's even more. There's this process of crossing over, okay? So there's even more opportunity for genetic variation to get introduced. So crossing over is another way the genetic variation can be introduced during the process of meiosis. And essentially all the crossing over is early in meiosis, before this first alignment. So you can see this sort of step here that's outlined in blue right here. This is crossing over. And basically chromosomes come together, like you can see right here, where it's sort of like the knees of the chromosomes are touching, and they can swap little pieces of DNA. So you can see here in this third step right here, there's this little piece of red that's become blue and this piece of blue that's become red. And so pieces of the chromosome have been swapped over, and that's called crossing over. And essentially what that ultimately results in the gametes that are produced at the end of meiosis 2 is you get new combinations of alleles that were not present in either parent. So you'd have this new red allele that's got a little bit, or I'm sorry, red chromosome that's got a little bit of blue in it, and blue chromosome that's got a little bit of red in it. You can see these little nubs down here with some blue and red and some red and blue. And so you've got brand new combinations of alleles now that weren't present in either parent, so even more variation, okay? So crossing over between homologs basically results in even more genetic variation in these daughter cells on top of that allele mixing from independent assortment and fertilization, okay? So just to sort of summarize in how meiosis and fertilization create genetic variation beyond just mutation alone, you've got independent assortment during meiosis, where those chromosomes can align independently, resulting in a lot of different combinations of chromosomes in these alleles that are, these gametes that are created. You've got different combinations of alleles that can come together in this diploid zygote from these two haploid gametes coming together and them having very different combinations of chromosomes in them. So once again, more different combinations of alleles because different combinations of chromosomes are brought together from mixing gametes during fertilization. And on top of that, sort of prior to this meiosis 1 splitting part, you can have chromosomal material crossing over, resulting in even more different combinations of alleles from those little chunks of chromosomes crossing over. So the sort of take home here is that all of these processes result in offspring with very unique combinations of chromosomes that weren't present in the parent cells and the parent organisms, and overall greatly enhanced genetic diversity in those offspring, okay? And this is really important to have that genetic diversity, to have the variation in the population that natural selection needs to operate.